All right, so let's talk about gametes and fertilization. So during the lesson, we are going to talk about meiosis in male and females. Then we will talk about the structure of the mammalian egg and sperms, and the sperm capacitation as well as the egg translocation, and finally fertilization and zygote formation. You should already know about meiosis, but let's talk about it so we can refresh our understanding of this process. But also we can talk about the differences in meiosis between males and females. Now, meiosis is a special cell division that occurs in specialized cells in the vertebrate body. These cells are called the primordial germ cells, and they divide by meiosis to produce haploid cells, which are the gametes. Now, during meiosis, the normal set of chromosomes, which is 2, and so cells in vertebrates are diploid, so they have 2 sets of chromosomes. So during meiosis, these 2 sets of chromosomes are reduced by half, so they become haploid end cells. So the first step during meiosis is the synthesis of DNA. So here you have cells after a synthesis of DNA, and during the synthesis of DNA, each chromosome is copied. So at the end of the synthesis of DNA, the cell becomes tetraploid. So they have 4 copies of chromosomes. So here, for example, you have said that this blue chromosome, this blue line here represents the chromosome free from dad, and the red line represents the chromosome free from mom. After DNA synthesis, you can see that this line is represented twice. So we have 4 copy of chromosome free instead of just 2. Interesting, the 2 copy of the chromosomes that have just been synthesized form what are called the sister chromatids. So they align with the old copy of the chromosome to form the sister chromatin. This is very important because the sister chromatin of the same chromosomes then align themselves to form structures that are even visible under light microscopy. And during this alignment, they can exchange DNA by process of crossing over. So some DNA from the red chromosomes move to the blue chromosomes and the other way around. And the point where this exchange happens are called chiasmata. Now, after the crossing over, so after the exchange of DNA, the chromosomes, the duplicated chromosome, align on the spindle, which is a protein structure that is made from the centrioles. This protein structure form a web, and the chromosome align in the middle of the spindles. So you can see here the chromosomes that are duplicated are aligned in the middle of the spindle in what is called metaphase 1. It is called metaphase 1 because in reality, meiosis is like 2 mitoses, one after each other, without an intermediate synthesis of DNA. So we have a meiosis 1 followed by a meiosis 2. So during the meiosis 1, the chromosome align in the middle of the spindle during metaphase 1, separate toward the 2 opposite centrioles. And they fall during the anaphase, they move at the opposite side of the cell. At this point, we have what is called the telophase. The telophase form a membrane that separate the chromosome that move on the opposite side and toward the centrioles, forming 2, indeed, diploid cells. But instead of finishing, meiosis continue in meiosis 2. And again, the chromosomes in each of the daughter cells align to new spindles originated from new centrioles. And this time, during metaphase 2, the sister chromatin are separated and move toward the different centriole in anaphase 2. At the end of anaphase 2, we have the formation of another membrane, so another telophase 2, separate the four daughter cells, which are at this point haploid. So after metaphase 1, we have the formation of diploid cell, but at the end of metaphase 2, we have at the end of meiosis 2, we have the formation of haploid cells. So this is exactly the process that is happening in during the formation of spermatozoa, so the male's reproductive cells. But it's not exactly what's happened during the formation of the eggs, and we will see that in a minute. So let's talk about the formation of the spermatozoa, which are the male's reproductive cells. This process is called spermatogenesis. Spermatogenesis start to occur during puberty. So before puberty, obviously, this process is quiescent, so it's not working. Obviously, the place where spermatogenesis occurs are the testes, and if you look inside the testes of mammals in general, so they're all very similar to each other, you will find a lot of tubules, and these tubules are called seminiferal tubules. And if we look a section of the seminiferal tubules, we found a number of different cells from the external part of the tubules releasing spermatozoa in the lumen of the tubule in the middle. And which cell do we find? We find spermatogonia externally. These spermatogonia are diploid cells that are progenitor of the spermatocyte. We have spermatocyte 1 and 2, which are the spermatogonia that initiate meiosis. Spermatocyte 1 are the spermatogonia that initiate meiosis and are still diploid cells, while spermatocyte 2 are the spermatocyte 1 that finish meiosis, and now they are haploid. These spermatocytes then go through a process of changing shape to form the spermatids. The spermatids are almost mature spermatozoa that then, at the end of maturation, are released in the lumen as mature spermatozoa. The spermatozoa include a head. The head of the spermatozoa has a nucleus with the DNA and a chromosomal vesicle, a midpiece that contains most of the mitochondria, and a long flagellum or tail. So, if we look inside the seminiferous tube, we recognize different phases of the maturation of the spermatozoa. We have an initial proliferative phase. The proliferative phase is recognized in the spermatogonia. So, at puberty, pulses of retinoic acid induce the spermatogonia to reproduce by mitosis. So, they reproduce by mitosis. So this means that the spermatogonia divide, they are diploid cell and produce diploid cell. Some of the spermatogonia at one point initiate a meiotic phase. So they start to reproduce, but this time by meiosis. And in fact, we can recognize the spermatogonia that become primary spermatocyte. These are diploid cells that start meiosis 1. And we can recognize spermatocyte type 2 or secondary spermatocyte, which are at the end of meiosis. They are already haploid cells and are the ones that then will change shape in post-meiotic shaping so that they form the spermatids. The spermatids are almost mature spermatozoa that at the end of maturation are released as mature spermatozoa in the lumen. So this maturation of the haploid spermatocyte 2 into spermatid and then spermatozoa, how does it look like? Well, this process is called spermiogenesis, and we can summarize this in this picture here. So this is the spermatocyte type 2 at the end of meiosis. You see it is very globular as a cell. We can recognize inside the mitochondria. We have a centriole that is important for the formation of the tail. We have a nucleus that contains the DNA, and we have the Golgi's apparatus. The Golgi's apparatus is very important because it will produce the acrosomal vesicle and granules. Now, the acrosomal vesicle and granules are fundamental for hybridization with the egg and therefore fertilization. So if this acrosomal vesicle is not produced or it is broken during production, then fertilization cannot occur. So what we can see during the process of spermatogenesis is that the Golgi apparatus produce the acrosomal vesicles, the mitochondria start to migrate toward the centriole, and the centriole produce a flagellum. This flagellum is made mostly of tubulin, protein. So when the Golgi apparatus finish to produce the acrosomal vesicle, the Golgi apparatus is discarded, and most of the cytoplasm of the spermatocyte of the spermatogonia is also removed and discarded, but some cytoplasm remain, change shape, cover most of the flagellum that is produced by the centriole until we have the formation of the mature spermatogonia that is released in the seminiferous tubulus. However, what happened at the end of this process of maturation is that we have a very long tail in the spermatogonia, in the spermatocyte, which is made mostly of tubulin, covered by a thin layer of cytoplasm, a middle piece where we find most of the mitochondria, and then a head where we find a nucleus and the acrosoma vesicle. Now, the spermatozoa are released in the testes, in the seminiferous tubes, and then they are collected in the epididymis. When the spermatozoa is released in the female reproductive organ, however, it doesn't have the capacity or the ability to fertilize the egg. So it acquires this capability through a process of capacitation that only occurs inside the female's apparatus. So before we talk about the process of capacitation, I'd like to talk about oogenesis, or if you want, the formation of the egg.